Yeah, so it's a pleasure to have Oliver give a talk here. Officially, he's Olivier, but I always knew him as Oliver. So I have to explain why I'm introducing him, because I'm not a biologist. So we met in 1979. That's a long time before many of you were born. We were graduate students in Caltech. We both joined in 79. We both graduated in 83. I think his defense was 1 week before mine, so he beat me to it by 1 week. We shared an office for a year, so we were good friends, and we kept up that contact for since then. Of course, after PhD, we diverged. So he did his PhD with Jeffrey Fox on lattice gauge theory. His undergraduate was in France, and after his PhD, he went. So that's high energy physics. Then he went on to Columbia University to T.D. Lee's group, lattice gauge theory. Then after that, his next postdoc was at the University of Illinois at Urbana, and by then he had moved to condensed matter statistical physics that was Kogood's group, Urbana. Then he went to City College, where he was a faculty member, and he started working in statistical physics. So that was the US part of his education. Then he went back to France, I guess around 91, and he joined the University of Paris, Orsay. Okay. And then he was working in statistical physics and spinning glasses, and he also co-founded an institute which he coined LPTMS, the Laboratory for Theoretical Physics and Statistical Models, where he was working with spin glasses mainly. And apparently Parisi was very active, and he collaborated with, he has a PRL with Parisi, I just found out. It was one. So after that, he decided again to switch to biology. That was around 2010. And I guess around that time was when Arijit started working with him as a postdoc. So he worked in genomics and Boolean networks. And that is for 10 years. And then in 2020, he got a full-time position, research position in this INRAE, where he's the director of research. So from high energy physics, condensed matter, statistics, biology, he's covered all the areas. So we have and now he's going to talk about multivariate challenges in mathematical genetics. I should say he's here for 2 weeks. This is his second week here. He's on a project with Arijit. So it's a grant. So let's hope he comes again on this grant. Okay, so
Thanks. So I'm delighted and actually even excited to be here. It's my first time. Hope there'll be a second. We're trying to plan that a little bit. I'm also very grateful for the really warm welcome I got, in particular from Bala, who's a long-standing friend, and from Arijit, who persevered to get us a CIFIPRA grant, and so that allowed me to come here this time, and I should be able to come again. So, as Bala mentioned, I've had a rather torturous series of topics on which I've worked on. As graduate students at Caltech, they told us theoretical physicists can do anything. Actually, it's not quite true. It was physicists can do anything. But okay, it's true that theory opens up to a lot of different fields. And so I worked for a long time as a professor of physics at the University of Paris, which has changed its name, but that's not very important. And I actually now am full-time researcher at INRAE, which is a governmental agency for research oriented towards agronomical type applications. And in particular, one of the initial missions was to help French agriculture after World War II. So this is an example sort of the kind of missions that we have. We want to help farmers develop better seeds or better plants for in terms of economic yield, etc. Okay, so it's going to be a lot about evolutionary forces and population genetics. I realize that that's not at all your field, but one of the messages is that in this particular field of, quote, genetics, there's quite a bit of mathematics, which can be of interest to people who are sort of theoretical physicists or mathematicians. And I just wanted to show you that indeed there are a number of different topics which are well adapted, if you want, to our skills. So there are a number of different things that we need to cover here that are maybe a bit basic for some of you. So one is a little bit of vocabulary. So you know that our genes are organized along chromosomes. As humans, we have 23 pairs of chromosomes. So these pairs are because for every pair, we get one copy from our mother and one copy from our father. Okay. And if I look at these genes, there are variants. So if I compare two homologs or just anybody in this room, there's a basically 99.9 similarity between two chromosomes number 4, for instance. But if I compare chromosome number 4 to chromosome number 7, there's essentially no real homology. And then there are variants for each of these genes. So a simple point mutation might be just that there's a base that's changed. If you have more major changes like deletions or insertions, that can lead often to disease. And so in particular, some of these allelic frequencies can reach a few percent, even though they're deleterious. Among the different evolutionary forces, we're going to focus on selection and genetic recombination. Why? Well, because I was in this plant genetics lab for several years. And in order to have this kind of genetic improvement that's mentioned here, genetic recombination, which is the shuffling of the alleles, plays a major role. So we're going to be, in this case, we're interested in trying to improve the value of a crop or the fitness of a particular individual. At the level of all these higher order organisms that are multicellular, we have sexual reproduction. So I'm not going to be talking about recombination in bacteria or in viruses because the mechanism is very different and the statistics is also very different. But basically, meiosis, you have then a diploid organism here. So we have 2 copies for each of our 23 chromosomes. We produce haploids, that is, these are cells which have a single copy for each of the 23. And then these 2 gametes, male and female, are fused to make essentially then the zygote. And the zygote is the single cell which is going to divide and produce then, according to development, the entire organism all the way to adulthood. And then you've completed the cycle. So meiosis, if you want to remember one thing, it takes a diploid cell and produces a haploid cell. Actually, it produces 4 such cells, okay, for various reasons. That's the strategy that nature has chosen. And the origin of meiosis is still a little bit mysterious, but clearly has a lot of components that are common with mitosis. What we're going to be interested in particularly is this the presence of crossovers. And so when you do this passage from diploid to haploid, you take a cell, and then we're going to make then these haploids, which have a single copy. And that introduces a little bit of shuffling, because if I take the 2 copies of chromosome number 4 that you have, you're going to pass on to your first child only one of those 2. Okay, so there's a little bit of mixture that arises there. We're going to be more interested in this type of shuffling, whereby within chromosome number 4, what you have on your father and your mother's chromosomes are going to be mixed, and that's associated with what's called a crossover. So this is the representation, if you want, of the chromosomes, and at the end, when you create these 4 gametes, they're going to be mosaics of the parental chromosomes. Okay, and that mixture is the key to all the initial developments in genetics. The objective there was to try to associate a given trait, and fruit fly was the major model system used there. A fruit fly that may have red eyes, well, what is the gene that's responsible for that? By doing these crosses and analyzing the descendants, you're able to basically create a map, what's called a genetic map, of the location of these traits or of these genes that are responsible for the traits along different chromosomes. So I've talked about crossovers. If you listen to people who are in genetics, they'll talk about recombination. Okay, it's almost the same thing, but for purposes of vocabulary, it's important to have the distinction. If I have these chromosomes and I have a crossover between 2 positions, and so the word position is replaced in genetics by loci or locus, you're going to be able to create individuals here whose chromosome is a mosaic of the parental chromosome, and that's called recombination. If I have just two different positions, then I have what's called recombined the alleles. If I have a mixture of blue and red at these two positions. Okay, so this introduces the shuffling that's of interest to the breeders.
So order of the allele is like one gene, but it's not always one gene that is responsible.
Okay, so this is of course how genetics began. One gene equals one protein equals one function. Of course, today we have a systems biology type approach.
But if they're doing if they're responsible for the same trait, so they're spatially correlated or close by?
So what we know is that the genomes are sufficiently mixed up that in general, if I consider two different genes nearby each other, they usually are in completely different functions. So there's very little correlation. In contrast to bacteria, the few cases where there's still a strong correlation tends to be in what's called clusters of genes associated with a metabolic pathway. Okay, so there's some structure, but otherwise you can consider that the genes are positioned with no particular logic. Okay. All right, so I mentioned the practical interest for breeding programs. And so let me give you a little sketch of what the different topics are that we're going to cover now. So the first topic is associated with how, for instance, selection and recombination or crossovers affect the dynamics of different genes in a population. Okay, so if you have a particular disease, how is that disease maybe going to be slowly removed from the population if it's deleterious? Okay, and I haven't actually published anything in this particular field, but I just wanted to illustrate it because it's there's some recent work by some colleagues at the École Normale in Paris that have sort of enhanced a little bit some of the previous work of Barton and Turelli. Most of the talk is going to be more on Fisher's infinitesimal model. If any of you have any doubts, this is Ronald Fisher, has nothing to do with Michael Fisher. Okay. Lastly, we're going to consider a number of different things which we can do a little bit more deeply in mathematics and in algorithmics because of the way the crosses are organized. And so these are called recombinant inbred lines, and that is work done in particular with Adigite. And so depending on how much time I have, I will cover these different cases. And so the first case will have what's called no genetic interference. And that's typically associated with Mr. Haldane, who was a geneticist, but has a close tie with India, as I discovered from Adigite. And then when you add interference, then the mathematics gets a little bit more complicated, and that's a project that we're still finishing with Priyatosh. And I learned, thanks to Bala, that actually Kosambi, who was one of the people who worked on this kind of genetic interference, was an Indian scientist. So this is then just a sort of a bird's eye view on what happens in the dynamics of populations. And as I said, this is not something which I have done anything worth publishing on, but the two main characters in this field are Barton and Turelli, who were able then to make significant sort of analytic progress on understanding what the roles of either selection or recombination have on what happens in a population. So they always treat the biologically relevant case, which is that you have a diploid. So the diploid means you have two copies of chromosome number 4, for instance. So I'll call haplotype the list of the alleles associated with a given chromosome. And so you get one from your mother and one from your father, so we'll denote it that way, okay? So the issue is how can evolutionary forces change the makeup of these kinds of populations? And so the treatment of a single locus, that is a single gene, if you have a disease, you can try to treat it as a single variable problem. But in general, things are multifactorial. You have to take into account multiple genes, okay, for any given phenotype. So, well, I'll be quick here, just simply say that in their formulation, they actually work with moments rather than the actual probability distribution. And if you have binary variables, that is, if your alleles, if you take into account only the possibility of having just two alleles instead of more than that, things get a little bit more easy to treat mathematically because of this property than if you consider that distribution is specified by all of its moments, arbitrarily high moments, then there's actually just a finite number of moments in the case of these binary variables, okay? And then you can write the recursions in terms of these moments instead of the probability distributions. So you are able to write those, and if there are not too many loci, you can maybe treat two loci analytically. So there's an entire book that considers 2 and 3 loci effects. And then when you go beyond that, well, things get a little bit unmanageable. So there are different computational codes for doing that. And at some point, you also have to get a program to write the code that will generate these probabilities or recursions, okay? So it's a little bit messy, especially because of the recombination. And so to obtain insights, people, in particular Barton and Torelli, have introduced either truncation schemes or what's called the quasi-linkage approximation. And the reason for having this little digression, because it is a digression compared to the rest of the talk, is that there's recently this paper on arXiv, which is co-authored by 2 people I know very well, Monasson and Coco, at the École Normale in Paris. And so they say that actually by going to cumulants, if the fitness function has a certain form, that allows you to go quite a bit further in the mathematical analysis. And this is built on the previous work that they've done. There's an entire community of people in statistical physics who are trying to treat what's called the inverse Ising problem, whereby if I have an Ising model with a bunch of Jijs that you don't know a priori, can I reconstruct those Jijs if I'm given the first moment, so in particular the average of each spin and the correlations, okay? It turns out that that's actually a technique that's used now a lot in protein inference. And so these people, they use that framework, and then they quantitatively check by simulation how well their different approximations work. And so what they've been able to do is, even though this is a quite old problem, they've been able to improve over this quasi-linkage equilibrium approximation. So for some variables, actually the predictions of all methods work quite well, but in the context of some other quantities, some are much more sensitive to that approximation, and what they show is that they really have led to an improvement in the method. Okay, so now we come to Sir Ronald Fisher's infinitesimal model. So this part is work that I've done in collaboration with my student, who's now a full-time researcher. This was published in 2024. And it turns out that in my lab, my genetics lab, my initial director, who's a quantitative geneticist, was a lot interested in the history of genetics and quantitative genetics in particular. So I was a little bit sensitized to these issues. So there was a great debate. So remember that Mendel's laws in the time of Mendel were never really diffused. And so it took until about 1900 for those laws to be rediscovered. And so there was a school of thought whose leader was William Bateson, who really had great success in showing that certain diseases and certain particular traits were strongly Mendelian. That is, if you look at the descendants, the laws of Mendel apply perfectly well. Okay, so that is a scientific success, that's fine. But in parallel, and even before all this, because as I said, Mendel's laws were not known for quite a while, there was a school of what's called biometricians that had empirically understood that there were some laws of inheritance. And so in particular, they were mainly interested in what's called continuous traits. So whereas the geneticists, they were interested typically in single gene type effects. If you look at any complex traits, so it can be body height, body weight, it can be muscle mass, all sorts of indices that are continuous variables, then the Mendelian approach is not very clear because, you know, when you treat with peas, whether they're wrinkled or not wrinkled, whether they're one color or the other, it's sort of a binary choice. Whereas in a lot of traits that are of interest, for instance, for breeding, the traits are continuous. So in this school of thought, basically there were some empirical laws. And so when you have, for instance, 2 parents, they understood by looking at a lot of different couples that the average height, for instance, is strongly inherited. The average height of a child will be the average of the 2 heights of the parents. Okay, so These two schools of thought were like two different paradigms. They had trouble communicating. Comes Mr. Ronald Fisher, who in 1919 was able to do a synthesis to show that actually if you assumed that it was not monogenic, but if you had multiple genes, you could go in some sense from here to there, and everything was coherent. Okay, so this particular paper is rather mathematical, but it took a while for it to have an impact. Clearly, Fisher was an extremely bright person. So today we still use Fisher's test or Fisher's information matrix. So his name is everywhere, and so he really is one of the key founders of these three different disciplines. So we're going to be interested in what's called directional selection. That is how things respond to a particular pressure for instance, greater body weight or others, okay? So there are many examples that have arisen naturally within the context of natural selection. And what's really quite impressive is that it's possible to do this in an accelerated fashion if you put enough pressure. So the breeding of dogs, for instance, was something that Darwin was very interested in. And so you can really shape a dog breed to have the components that you want, short tail, long ears, etc. For any of you who have one of these purebred dogs, you know that along with the breeding basically makes them homozygous. They are very susceptible to all sorts of problems. Okay, so I have a friend who says he needs to spend over $1,000 every year going to the veterinarian because these animals are generally quite sick. Okay, so this poor chicken is going to give you a big biomass, but it's also very, very unhealthy. Okay, so if we come to plants, which is simpler for me, I'm happy to cut plants down. That's not a problem for me. My lab worked a lot on maize and on wheat, and what we see is that there has been a continuous progress in the genetic value. So the genetic value here typically is the yield of these different crops over the past century. So there's a near linear growth, and so that's not completely independent, of course, with the Green Revolution. If you just go ahead and say, I'm a theorist, let me try to do some modeling associated with that. You can see empirically that within a reasonable genetics model, you're going to get such kind of improvement by going to get the alleles that are favorable for your particular trait. We were actually interested in whether if you increase the recombination rates, you can speed up the improvement, the genetic improvement. And so that's what these different curves are. So what you can say is that increasing recombination helps, in fact, not only improve the average value, but you're going to maintain a little bit of the selection on other traits. And so today, if you want to breed, it's better to do it a little bit more slowly and to also make sure that the genetic diversity stays there. Okay. So Fisher's infinitesimal model, in its simplest version, you simply say that you have a underlying genetic model with genes. Okay, so we're going to label the genes A, B, C, etc. And we're diploid, so every single one of these genes comes within 2 copies, and you can have 2 different alleles eventually. Okay, so what Fisher says is that the genetic value, which means actually, for instance, something like the height, a continuous variable, is going to be just a linear combination of these different allelic values. Okay, so that's the additive model, which is really the simplest thing that you can do. And if you talk to a geneticist, he will say, yeah, we're forgetting all the dominance effects, you're forgetting epistatic effects, but we're mathematicians, so we say we're going to make some progress on the simplest model. Okay, so in his simple model, he takes what's called the infinitesimal limit. So you take an infinite number of these genes, okay, and each one is going to contribute infinitesimally little. Okay, so what you can show is that if the initial condition is Gaussian, then after 1 generation, you're going to exactly find the properties that the biometric school likes. That is, that the children will have the values that are Gaussian and centered on the mid value of the parents. So almost everybody's happy, but when there are 2 different paradigms that are in competition, the people who have been brought up in one paradigm always psychologically have difficulty accepting the other paradigm, okay? And so there's an old saying that theories don't take over. It's just that theorists or scientists retire. Okay. So one thing that they said, no, this is not reasonable in terms of the proposal of having this infinitesimal model is that if you think of taking a series of pigments, and so these pigments are like the genes or the alleles. If you say that from one generation to the next, you're going to start combining them, well, at the end, you just get one brown color, essentially, right? And it's not very difficult mathematically to show that that's incorrect. If you just take this model of Fisher and you say that every single variable here has mean zero and has a variance going like 1 over 2n, then the overall sum will just be a normal variable. And if you just keep on sampling from that population, it will be a steady state and will perfectly well maintain the variance. Okay, so it doesn't go to everybody has the same genetic value. Okay, so the question then, nevertheless, is well, what happens if I do some selection? So you have a population with some alleles which are favorable, for instance, for height, and other alleles that are not favorable for height. Okay, if you do selection for higher height, you're going to enrich in those alleles that have the positive values for your height. Okay, and so after a while, you will have done your enrichment. There's no more enrichment to do. Okay, the magic of Fisher is that there are an infinite number of such alleles. That is, you have a reservoir which is infinite. So you can pull out from that reservoir favorable alleles, but you're having an infinitesimal effect on the reservoir. So you can keep on going. And so there will be basically no limit to the amount of genetic gain that you can obtain. So schematically, this is the way it works. You have a distribution, for instance, for this quantity G, which is the sum of all the allelic contributions. Experimentally, what one does is one simply selects all the individuals above a certain value. And from generation to generation, you hope that this will lead to a genetic improvement. So what I've done here is I've taken, indeed, the way people operate in practice. This is what's called truncation selection. I select all the individuals above a certain value. Their descendants, they're going to have then an improved average value because the model says that the children have, on average, the average value of their parents. The parents are better, so the children will also be better. Okay, so you start with these parents, which are selected. You undergo then the sexual reproduction. You have the gametes. You produce the children. So the question is, after you've done this, will you have a variance that will be similar to what you had initially, or the fact that you've selected for good individuals, the mean will have shifted, but maybe the variance will have also reduced. Okay. So one scenario is yes, and it will turn out that in a particular choice of Fisher's model, you can show that that's the case. This infinite reservoir that I just mentioned is there, so you'll always have the same variance here. An intuitive point of view is that, well, actually somehow there's a diminishing variance, and so progress will slow down, and that would correspond to this kind of system. But that is associated then with the idea that as you improve your genetic value, well, there's less and less amount of allelic variation to go get. Okay, so that's the story for basically genetic selection in Fisher's model in the presence of basically what's called random segregation of alleles. There's no linkage. And we were interested in what happens when there is linkage. Okay, so mathematically, what we did is we came back to the model without linkage. We realized that actually it's a little bit simpler if instead of taking this truncation selection, which is introducing a completely non-trivial distribution on these values, you just say, I'm going to take a selection not for a whole continuum, but for just one value. Okay, so this Dirac selection you can do mathematically, of course, and that's going to simplify a lot the mathematics. Even within the original Fisher model without linkage, it allows you actually to rather simply derive all the results. And so in particular, one of the sort of the subtleties is that when you do this selection, because the reservoir is infinite, its effect is in some sense infinitesimal. So if I take any random variables that are Gaussian, multivariate, if I do any kind of constraint, which is linear, the resulting ensemble is still Gaussian multivariate. Okay. One of the sort of the magical things is that if I do this with an infinite number of alleles, that selection has, in some sense, a negligible effect on all the variances. The variances stay the same. The covariances remain negligible in quotes. And the mean has changed. So you have this initial ensemble, you do the selection, the mean changes, but in fact, all the covariances are hardly changed. But that hardly, there's a little subtlety. Okay? So at the end of the day, just as to summarize, once you do the whole reproduction and selection steps, you end up with the following system. You have an initial population of diploids. The variance by construction, we set it to 1. After selecting for a particular value of G, the variance is 0. But then there's some variance which is released when you produce the haploids. Okay, so a simple way to think of that is that if I have any number of parents which have exactly the same sum of their X's or their AIs, if I choose a subset of those, I lose that constraint. So I'm going to release variance. That is, I start with something which has a population variance of 0. I choose subsets of alleles, okay, and I lose the constraint that the sum of the X's is equal to 0, so I have some variance. That release of variance, there's this little argument which allows you to show that, in fact, that release of variance is precisely 1/4. So each gamete will have 1/4 of the initial variance that was here. And then you enter this loop and it's a steady state. Okay? Well, it's periodic, but so you start with this population which has finite variance. And at the end, you have a system which is alternating between variance of 0 and 1/2. Okay, so now the key word is we add linkage. Why do we add linkage? Well, because that's reality. We know that the genes are organized along chromosomes. So during then the reproduction stage, you're going to make mixtures, and these mixtures are not completely random. In the no linkage case, you just take all the different alleles and you just mix them as if there was no physical position along these chromosomes. Okay. So what we do know, of course, is that when we have these crossovers, then there are few crossovers for meiosis. If you have 2 points which are very close to each other, they generally will segregate together. So what that means is that the shuffling is ineffective for close-by genes. You're going to shuffle genes that are far away, but when you get close by, the genes are not going to be shuffled except very rarely. Okay, so that's going to slow down the process, of course. Okay, so we end up with 2 opposing scenarios. The first one, which is sort of the textbook scenario, which is that, well, mathematically, we don't know how to treat the system, but nevertheless, there's still going to be some allelic variation to go get. And so the idea is that the linkage effects will in some sense stabilize. You're going to go to a steady state. It's just that the gain per generation will be a little bit lower. Okay. And then there's a different, maybe more intuitive point of view, which is that, well, we know that we're going to want to go get this infinite reservoir. The infinite reservoir is still there. It's just that it's going to get harder and harder to access it, because you're going to want to go and get genes that are closer and closer together. Okay? So this is sort of, in some sense, what we get from this intuition. We still have to do the mathematics to show that that's, in fact, really what's happening. But when I talk to colleagues who were brought up with the paradigm, which is in the textbooks, they say, well, look, also, there's not just this previous result, which seems to be, a general result. But if you look at the empirical results, we see the linear behavior. Okay, so why you bother with such a problem? Okay, so first thing you do is you do some simulations. When you do simulations as a function of number of generations, you have to then, to approach Fisher's model, you have to take the number of genes to infinity, and you have to take the number of individuals to infinity too. Okay, so here's just sort of a schematic of what happens when you increase the number of individuals. What you see is that there's something like a limiting curve that's arising. And if you are then in the framework of the first scenario, you say, well, ultimately, this will go to a straight line. But you see that it doesn't exclude the possibility that there's some kind of curvature in the system. And you can imagine that the curvature is also something that's reducing to going towards zero. Okay. So my conclusion, mainly a numerical person. So my conclusion is the question you're asking here are just too subtle for numerical analysis using these kinds of simulations. Okay. So you have to treat directly a different framework in which mathematically you first take m and n to infinity, and then maybe you can do some numerical analysis, okay? So that's what we're doing. We're going to take directly the mathematics associated with m and n going to infinity. And so just to make the punchline short, so basically what we find is that indeed, when you do the directional selection in this particular way, then it is going to get more and more difficult to go access that variation. So the genetic gain per generation, as well as the variance in the population, will slowly go to zero. Okay. The difficulty is it goes to zero very, very slowly. So that makes numerics pretty tricky. And so in particular, it goes with the log, 1 inverse log of the number of generations. Okay. So that means that doing any kinds of numerics is a little bit subtle. To make the mathematics as simple as possible, we took some hypotheses about the way, for instance, the chromosome was organized. So instead of taking a linear chromosome for practical purposes here, we can consider the case of circular chromosomes. And the other thing is when we do reproduction, remember that when you have two different chromosomes that are homologs, they're going to exchange at random positions, more or less. And that means that the descendant may recover a little bit more one chromosome than the other. To make the mathematics simpler, we force then the amount that you inherit from each parent to be equal exactly 50%. Okay, and then there are a number of other things. We work with selection at the level of the haploids instead of the diploids, etc. Okay, so very briefly, you go into Fourier space to make things simpler. So this this x, which is the allelic value as a function of the value theta, then can be replaced by a list of modes if you want. So this is the complex component of mode number k for a Fourier analysis. You can then calculate the first moments, and the initial condition is very simple. Everything is a complex Gaussian. Okay. So then you want to say, I'm going to implement the iteration. So I'm going to select all the individuals that have a given value. And so from one generation to the next, you take in the curve. So if I have currently a distribution like that, so you could say, well, I'm going to go and get things that are arbitrarily far, but of course, from the practical point of view, that's not possible. If you are breeding things, you only have 1,000 animals, for instance, you cannot go arbitrarily far. So the convention is that you stop at some number of sigma. So you have a coefficient here, and you take, when you do truncation, you take all the individuals that are beyond 1 or 2 or 1.5 sigma, and we're going to take them the values that are exactly at that amount. So that's what this corresponds to. You take only the individuals that have exactly this value of G. So what that does is it affects the xk when k is equal to 0. And all the other modes are unchanged. Okay. So this gives you a recursion for the mean of this distribution, which just says that indeed, when you take all the individuals here, you know a priori what the mean will be because everybody has the same value. And then you need to do the second phase, which is the reproduction, which involves then these two crossovers. And that's where it's a little bit more tedious. You have to introduce this periodic Heaviside function to keep track of only 1/2 of your circular chromosome. That's what this does here. You take the Heaviside function and multiply it by your x. So you're going to have a profile, which is 0 on 1/2, and then the original profile on the other half. And this allows you then to continue the process all the way to combining the two gametes. And so this is the Fourier component for the child, which is diploid, which will be then the sum of these two contributions. But then what you see is this convolution. That is, all the different modes in different k values will contribute to this recursion value. So at the end of the day, you say, well, okay, I have a new x, which is a linear combination of the older x. And so if these initially had a Gaussian distribution, then this will also have a Gaussian distribution. So I was very pleased with that. But the problem is that this here depends, this distribution, and in particular the covariances, depend on this quantity phi. And so I wasn't able to go all the way and say that the overall distribution, when I consider all possible events, remains multi-Gaussian. So what I did is I projected, I said, I do like Barton and Torelli. We're going to consider the variance and covariance matrix and do the recursion on those quantities. Okay, so there's likely an approximation. When I discussed with the referee, basically I was almost convinced that I should consider that he was right. I'm not completely sure. It may be that some of this stuff is exact, but in the worst case, we simply say that we're dealing with this Gaussian approximation. That gives us these recurrence equations for all the different variances of these Gaussian variables, or these variables that are considered to be Gaussian. So what's important here is in this recursion is that the variance, and in particular the variance that we're going to be interested in, is for k equals to 0. That's the variance of the total sum of the xi's. It has a recursion which can be interpreted as a hopping process. So you have a Levy law. So the hopping decreases like 1 over k squared. And so that's like a Levy flight. And then you have at each iteration, you have a smoothing, if you want, of the profiles over k. And you have a conditional quantity here, which remembers what the previous generation was. G here is generation. Okay. So if we treat this quantity numerically, here's a function of the mode k. We're going to have the evolution with time or with generation. So G is the generation of this whole list of these xk's, or at least of their variances. Okay. Well, what we see is that what's plotted here is 1 minus sigma squared. So if we consider just the mode k equals to 0, we're interested in what's happening at this position. So if we start with small generations, we can follow the curve, the points here. And so ultimately the question that we're asking about the importance of linkage is, what is the asymptotic value of this little peak here? Is it going to go all the way to 1? If it goes all the way to 1, that means that this variance here is going to 0. And that will be then the second scenario as opposed to the first scenario. So the first scenario is that this will rise and stabilize at some finite value. Okay. So there were a number of numerical tricks here that had to be used, and in particular, I had to do some matching between the numerics and some analytical function form, because precisely it's Levy hops, and so that's very, very spread out. So for numerics, that's really bad. What you then say is that, well, I'd like to try to analyze the behavior with the number of generations. So everything was represented in terms of the log of the number of generations. And you expect a scaling law, okay? So in scaling, sometimes the scalings work out very nicely. For many problems which have slow conversions to scaling, you really need to include the first correction to scaling. So that's what we did. So at the end of the day, we found that if you did the following system, you say that basically the variance decreases roughly as 1 over log of the number of generations. You take the product of those two, that hopefully will go to a constant, but then the correction is to scaling. So, okay, if I only consider the constant, that corresponds to having this plot here go out to a constant. If I just show you this plot, no reasonable person would say that that's really good evidence for going to a constant. You really need to have some kind of curve which is telling you how you're converging eventually to a constant, okay? So we ended up with this functional form, and I have to admit that this took us a long time to find something that finally worked. And I was inspired by some of the results from Derrida, who had looked at different models of one-dimensional propagation and creation of wave fronts, where he had these strange logarithmic, or it's actually log of log corrections. So if it weren't for this term here, we would have found it very quickly. But this log of log term got us very much hung up, and it took a long time. But at the end of the day, so the curve, this analytic form is given by this. We do the fit for A and for B, and that gives us this quantity. And so you see that the numerics is really going towards this envelope. Every time you double the size of your system, you can gain one good point in this log scale here. And so this is numerical evidence that, in fact, indeed, your overall variance is indeed going to 0, okay? Now, it turns out that once I had convinced myself that this was indeed what the behavior of the equations were, I went back to the recursions and you can show mathematically that in fact it cannot go to a steady state. Okay, so that was relevant. Nice. Okay, so the conclusions is when you go from no linkage to linkage, it's kind of a singular consequence that you abandon steady state and you end up with something which is more like what people see in glassy systems. There's a slowdown. The slowdown is logarithmic. Okay. Okay, so I'm going to go very quickly through some of the cases of these riddles becoming inbred lines, partly because some of the work was done with Arajit quite a long time ago. So a riddle is a resource which is fair. So it's accessible and it's reproducible. So basically it's collections of individuals, so it can be animals or plants, which are homozygous. That means that when they reproduce, they give identical children to themselves. That means that you can share this genetic resource with any scientist in the world. And everything then is perfectly reproducible. You can say it's fair, although you can order these things so they can be shared. Of course, you have to pay, okay? There's a cost for maintaining these resources. So these are used a lot for mapping diseases in mice in particular, or rats. So we're interested more in the case where we're dealing with plants. So in that case, you can do single seed descent, which means self-crossing. So let me just remind you that most plants are bisexual. That is that in the flower, there's both the male and the female organs. That means that you can make seeds out of a single plant. So this part here, I'm just going to cover some elements on how you make these riddles. And so basically, this is work that was done with Arijit, and then more recently we decided to expand it to do work with Priotosh. So if I have two different loci, two different positions, then the recombinant haploids here are mixtures of these two chromosomes. So when you want to create these genetic resources, the idea is that you start with parents that are homozygous. You cross them, and at the level of the second generation, you're going to have variability. So every single individual here is in some sense unique because the positions of the crossovers are going to be unique from one generation to the next. And if you repeat this many, many different times, at the end, you end up with what's called the real population. So these inbred lines, they're completely inbred, which means they're homozygous. The copies, the allele on each locus is the same from one chromosome to the other. And that is the resource that you can use to try to map where the disease is being produced or causal. So with Arijit, we looked at what happens in the multi-locus statistics. So it turned out that Haldane and Waddington had solved this problem for 2 and 3 loci. We showed that by using a number of tricks from statistical physics, in particular Schwinger-Dyson equations, you could actually derive self-consistent equations, which allowed you to actually solve this problem for, in principle, an arbitrary number of loci. So this is just the mathematics associated with when you have 4 loci, and there's also a nice graphical interpretation associated with these different diagrams, okay? I'm going to stop with this particular formula. The idea is that when you have these discrete spins, because they're just 2 alleles, everything is biallelic, you can have a finite number of moments. And so in particular, if you want to describe what's happening with 4 loci, you need to calculate, or you need to have the values for the first, second, third, and fourth order moments. And that's what this quantity is. And what's nice in the Schwinger-Dyson equation is that this moment here is related to moments of similar or lower order. So given that formula, this formula, as it's written, allows to treat the arbitrary case of with or not interference. With Arijit, we did the case of no interference. And with Priotosh, we're finalizing the case with interference. And so genetic interference is the fact that the crossovers are not independent. Okay, so if you have independent events, that's what's called Haldane's model. If you look at the distance from one crossover to the next one, you have just an exponential dependence. Whereas with interference, things have, in some sense, a deficit here at the level of nearby. So it's as if basically crossovers were repelling each other. Okay. So as I said, this is really what's happening almost all the time. Almost every single species will have this little depression in crossover distances. So in terms of modeling this, one of the most popular models is what's called the gamma model, which is a kind of stationary renewal process. And what we did is that we considered one particular case of this gamma model, where this parameter here, which is associated with this gamma law, is an integer. There's a nice interpretation of that, which is why people in genetics called it the counting model. That is, you can say, I'm going to do an exponential process once, twice, m times. And after m times, I say I have my new crossover. So that allows you to get this kind of dip or this deficit at small values for these crossover distances. We start with exactly the same equation for the mathematics. It's just that now these probabilities for having given haplotype at the gamete level depends on this parameter n. Okay, so once you have that, you can use very similar codes. We built our code with interference based on the code without interference. A lot of the parts were in common. And then in terms of algorithmic performance, then you can get all the different probabilities for these rules for up to about 10 loci and calculate that on a desktop computer. Okay. So you can do description of these different parameters. And so one thing that a lot of people in genetics use is what's called the coefficient of coincidence. It's sort of comparing the frequency of having recombinations in multiple intervals to what you would expect if there was no interference or in some null model. And so what you can see is that this is going to allow you to discriminate between data that is normally described by a particular value of m. So for instance, this value of n compared to other values of m. So we're going to be able to use this for inference. Okay. So these probabilities, you can use them as likelihoods, but you can use them also as summary statistics. So that's what we've been doing with Pirtosh, and then there's various optimizations to do on how you choose these positions. For instance, when you calculate these multi-locus events. Okay. Then lastly, let me close on the case of what's called SIB reals. So everything with single seed descent is only relevant for plants. When you have animals, sexual reproduction. Okay, so these mice here are going to be then the example. So at every single generation, you're going to need to have both sister and brother to be able to maintain the system. So you have these iterative sort of process, and at the end, you get inbreeding, perfect inbreeding. Everybody is homozygous. Okay, so again, interestingly, Haldane and Waddington had done the SIB case for 2 and 3 loci with a associated formula. But again, since 1931, there was no results beyond 3 loci. So with Kamel and with Marianne, we then said, okay, we want to try to do the case with more than 3 loci. And it turns out then that the mathematical formulation that we use with Adijit is not at all appropriate. You end up with 16 to the L different quantities to follow, and it's just not tractable. If you use instead something which is called identity by descent, there are only 4 to the L quantities. And so that allowed us to then say exactly, we can generate similar equations to the Sherman-Dyson, but with these other quantities. So to be very brief, these identity by descent is something that people in genetics use quite often. It says simply that if I look at any given piece of DNA in any individual at any generation, there's a certain probability that that DNA comes from one of these 4 quantities, one of these 4 chromosomes. Okay, so if you write that, there are exactly 4 to the L different quantities that are unknown. And there are a number of equations that we wrote. And at the end, you end up with just a purely linear system. Okay, it's a bit of a mess, but it's a linear system. So in principle, you can then just do matrix operations to reconstruct exactly all these 2 to the Q to the L quantities. And then that gives you all the different probabilities that you're interested in. So the overall procedure, the flowchart is quite a bit more complicated than in the work that we did with Adijit and Priotosh. And at the end, you can then use these computer programs to look at different statistics. So one of the things that people often do is they do their genetic mapping by saying, well, we don't know what the probabilities are. We use what's called the independent interval approximation. What you see is that generally that approximation is in fact quite good. Okay, and then you can ask, well, if I put in interference, because this framework also allows you to do interference, does the interference do very much? And you see, well, interference does a little bit, but it's more like 30% effects than factors of 2. Okay, for a biologist to be interested in anything, there has to be a factor of 2. Okay? All right. So in summary, basically, there are a number of different mathematical problems that you can treat with techniques from statistical physics or more advanced mathematics. And there's still quite a number of genetic problems, mathematical genetics problems out there that are still looking for either better approximations or exact solutions. So I'd like to thank my collaborators. So there's Arijit and Priyatosh here at IMC, and then at the University of Paris, different people who are postdocs or collaborators or students with me. Then all this has been possible to show you thanks to Sefipra. So with Arijit and a little bit of his insistence, we finally got a grant from Sefipra, which is funding me to come here. These are different funding agencies of Arijit. I belong to a network that funds overall plant science in my area. And Priyatosh came to visit me for 3 months on one of these Raman Charpak fellowships, and we are all together then to try to finish this project. So thank you very much.
So, in the second part of your talk, you presented that model with the logarithmic corrections eventually. And you said that eventually the variation does go down, and therefore presumably the gains from
so the gain per generation goes to zero.
Goes to zero.
Okay, so it was on my slides, but I didn't sort of insist on that. There always is this reservoir, infinite reservoir. So if you're patient enough, you're going to access it. So the total gain is not bounded. You can go to arbitrary gains to show that it's very, very slow. It's logarithmic.
So then, you also mentioned this empirical result, which is that people don't really find a slowing down of gain. It keeps going linearly.
Yeah.
So how do you square the two then?
Okay, so this is the real world. Okay, my model is an ideal model. There's no mutation. And presumably, there have been some experiments of this type of directional selection in maize where you start with a homozygous line. You just take one line that's homozygous. So there's no genetic variation. And they see that there is a trend. So the trait that they looked at was flowering time. Because of climate change, we know that the flowering time is going to have to be adapted to the local environment. Okay, and so what they were able to show is that you can do a selection for earlier flowering time with a starting individual that has no variation. And so it's all about a new variation coming in by mutation, and then you can say, well, mutations are pretty slow. How's that happening? Turns out it's mainly the transposons that are moving around.
I see. So that the models do not yet take care of that.
And I would not want to worry about that because what a mutation does is totally dependent on where that mutation is. It's you're sensitive to rare events. It's very difficult to treat extreme events in any kind of mathematical framework. That's relevant for biology. We'll put it that way. I mean, for me, biology is the science of extremes. You've selected for things, and so in any ensemble, you're looking for something that's extremely rare, and that's what the biologist is interested in. All the rest, he doesn't care. So the generic behavior of a biological system means nothing for him. Right. Things are not generic. That's the problem. In physics, we like generic things. Measure one, right? Nonsense.
A couple of questions. I didn't follow all the details of your talk, sorry, but one question is, I'll read the paper carefully. One thing is, how realistic is it to do detailed simulation of this, take a plant genome or whatever genome and simulate the meiosis, simulate the crossovers, simulate the transposons, mutations, whatever you have, and compare it with what your model predicts.
So if you're saying that you'd like to try putting in some mutations to see what can
I'm
happen.
saying, no, just take an in silico, population of plants and select and breed and all that in the computer
On the computer.
and see how traits, evolve and get selected.
So clearly, they had not anticipated that there would be such a response to the selection in this maze experiment. Okay, because they hadn't thought that, well, transposons can really rewire the regulation. Okay, so that's probably what's happening. If you want to try doing modeling of that, you could consider it a huge success if you could predict what the average rate of changes. Right? But I would say a more realistic goal would be to say, okay, let's try to estimate that rate within a factor 100. Because all this depends on how these transposons are reactivating and how quantifying their effects. And even the factor 10 seems unrealistic.
And the other question is more, practical, since you're talking about, benefit of plants and so on. And you mentioned that, purebred dogs are unhealthy and, what's the trade-off in, breeding rice or maize or whatever for desirable traits versus robustness to disease? And is it okay? And I think this question is probably relevant to India and other countries.
Yield is a very quantitative trait. Okay, that omni-gene, there's probably thousands of genes. When you do these GWAS analyses, you find that there's thousands of sites that potentially contribute, but a very small amount to any given trait. So it can be the height or the mass of the seeds, etc. When you're talking about disease, then the breeders are really interested because there is typically just a few genes. And that's within reach. And so a lot of these companies, they mainly focus on resistance genes. That's the thing. So the genes actually turned out to be often clustered, and there's kind of like a war going on between the pathogen and the defense system. And so plants don't really have an system the way we think of it for animals, but they have all this complex machinery to try to actually prevent or defend against pathogens. So either by recognizing the pathogen before it is that able to penetrate the cell, or if it penetrates the cell, there's a mechanism of suicide. So the cells can commit suicide apoptosis in response to certain signals. And it's all about sort of an arms race, just like for viral infections. So you have to have a part of your genome, and it's going to be in the cluster in which there are transposons that is very labile and is going to be able to react or produce variation, and that variation will be selected according to the pathogens in the environment. And so if you want to breed, so you have some rice that works well in the United States and you want to bring it here. So there's climate change and there are also different pathogens. So the idea is that you're going to try to first introgress some of the favorable alleles into the elite maize or rice that you have in the US. And then you can try to have a breeding program here that, given that starting point, you can still optimize a little bit.
I guess my question was more like, is there, and this might be more philosophical, but is there a possible trade-off between accepting 25% less yield versus being more robust to pathogens? Or, for example, there was this banana cultivar called the Gromichel, which was dominant a few decades ago, and it got wiped out by a fungus. I'm not talking about India, but internationally. Now the common banana cultivar is the Cavendish, and never that will also be wiped out. So here in India, you'll see 20, 30 different varieties, but those don't ship very well to other countries. That's this aren't highly inbred lines and also monocultures more susceptible to this kind of thing.
Yeah, so the inbred lines, especially in animals, they're all quite sickly, just like the purebred dogs that I mentioned before. In the case of plants, it's less clear, but it's known that there's much more redundancy, genetic redundancy in plants. So you can have multiple copies or paralogs of genes that have almost the same function. That makes genetics in plants both easier to do because you can change a gene, which in an animal would be a lethal change, and you can still have the reproduction, still the genetics of the crosses in plants, okay? If I come back to your initial question, which was about trade-offs, what's clear is that if you have a defense system against a pathogen or a class of pathogens, there's a little price to pay. Okay, so it will decrease your yield if you're robust against a large number of pathogens. Okay, so then the question of whether you want that, where you want to be in that trade-off, well, that's the bet that the farmer makes, right? So he has a choice of different seeds. He's going to say, I'm going to bet that this year there's not too much problem with pathogens, and he's going to plant that. And if he's wrong, well, he loses his whole harvest. So people now really, indeed, they're going towards robustness is a very important key factor, partly because with climate change, fluctuations are increasing, so there's less predictability.
The line that you choose,
Yes.
is there any other possibilities?
So,
Simple
so what you know, so more pressure you put,
Yeah.
so because okay, all this is modeling, right? There's an infinite reservoir. I told you. In the real world, the reservoir is not really infinite, right? We have a finite number of genes on chromosomes. Okay, so if you're a breeder and you want to have, for instance, horses that run faster, okay, there's a lot of money behind that. The horses, they're going to have to run faster, but maybe they also better not fall sick too much. So these kinds of compromises. Clearly, if you're in a hurry, you're going to put more selection pressure, and then you're going to say, okay, he runs really fast, but actually his lifetime is only 6 years, and then it's not a good investment. So the difficulty is that to have something which is going to work really well in the long term, you need to have large populations, and you have to select for less. You have to maintain the diversity. And so what's known is that the selection pressure really is more effective the bigger the population. If you have small populations, you're going to have drift, and some sort of the unfavorable alleles are going to be fixed in the population. That's just the drift.
So, if maintaining phenotypic variability is kind of interlinked with fitness, somehow, like I come from like kind of bacterial physiology, so it's always good for these microbes to maintain a sufficient variability to adapt for uncertain conditions. So, is it also true if you maintain a certain kind of yield, you also have to do, sufficient amount of variability in the phenotype so that it can face other adverse conditions.
Yes, so it's clearly a major factor in both robustness and adaptation. So when the farmer buys these seeds that are homozygous, or there's also a whole industry for building hybrids, of course. The point is that it is extremely high performance in certain conditions. And so basically this green revolution has been made possible by having plants that really use the fertilizers extremely well. So you have a system whereby you have like a F1 formula, a high performance car, but it can only run on perfectly smooth roads. Okay, so you can have very high performance if you're willing to sacrifice any robustness. In the case of the bacterium, they're going to think about their own interests in some sense. It's anthropomorphic, obviously, but for anything to survive in natural conditions, you have to maintain that diversity at a population level, at least. But at the individual level, it's also better to be heterozygous than to be homozygous. So in the case of bacteria, that's not an issue. But part of the success, if you want to higher eukaryotes, is partly perhaps because we're diploids. As diploids, so a simple example I like to give is if you have an enzyme, it's going to have an efficiency which is going to be low and low temperature just because of the Arrhenius factor. That low temperature things, the energy barriers are not going to be overcome. High temperature, well, the binding between the receptor and the ligand at high temperature, there's too much thermal noise. So there's some optimum at a particular temperature. Okay, So what does that mean? That means if you're not cold, warm-blooded, if you're cold-blooded like a lizard, it's better to have two enzymes, one that operates at 16 degrees and one that operates at 20 degrees. Okay, that way you're always more or less adapted.
Thank you.
Okay. No questions. Let's thank Oliver again.